Hello, and thank you for coming to check out my poster. My name is Amanda, and I'm a graduate student at Seton Hall University working under the mentorship of Dr. Michael Vigorito. In our lab, we have been focusing on a Pavlovian conditioning model, also known as sign tracking, as a way of evaluating compulsive-like behavior in Sprague-Dawley rats. So as a bit of a background, in a sign tracking paradigm, we have two important components: a rewarding unconditioned stimulus, which I may refer to as the UCS, and a neutral conditioned stimulus, which is the CS. Typically, the CS immediately precedes the UCS, therefore acting as a signal for the delivery of the reward. With these repeated CS-UCS pairings, two different behaviors tend to develop. Animals may choose to ignore the CS during its presentation and instead interact with the reward delivery site. This behavior is called goal tracking. Another possible behavior, known as sign tracking, is when animals actually begin to interact with the CS in anticipation of the reward UCS. Because the reward delivery is independent of CS interactions, meaning that the animal doesn't actually have to interact with the CS in any way in order to get the reward, sign tracking has been referred to as compulsive-like. In fact, it's often looked at as being possibly maladaptive. So past researchers have demonstrated that ethanol can actually serve as an effective CS. So the sign tracking paradigm has been used as a way of inducing ethanol consumption in rats. If sign tracking is truly compulsive-like, then this might shed some light on the compulsive nature of substance abuse and addiction. What we were especially interested in for this study were differences between environmentally enriched and standard housed rats on sign tracking behavior. A predominant idea is that environmental enrichment is a protective factor against substance abuse, but there have been very few studies that actually looked at enrichment within a sign tracking paradigm. The one Beckman and Bartos study that's cited in the introduction here is one of those such studies, and the authors found that animals reared in isolation displayed more sign tracking behavior than did animals reared in an enriched environment with the opportunity for social interactions. Our study differed from this model or this method in that we ran sign tracking for a total of 18 days per animal as opposed to only 5. Our study differs from every other sign tracking study that we are aware of in that we tested whether sign tracking emerges when testing occurs in the home cage rather than a distinct testing chamber. So just a quick overview of our method here. We had a total of 16 male Sprague-Dawley rats. They were assigned to either the enriched group or the standard group at 21 days of age. Standard rats were housed in pairs in typical laboratory cages, while the enriched rats were separated into the towers that you can see in this picture. We had a total of 4 rats per tower. At 5 weeks of age, we began testing the rats. All the rats were tested on the top landing of the enrichment towers, which you could see on the picture to the right. So enriched rats were tested within their home environment, while standard rats were removed from their cages and tested in this less familiar context. Each sign tracking session consisted of 25 trials of CS-UCS pairings. So for the CS, we used a sipper bottle containing 5% ethanol solution, and for the UCS, we used sugar pellets. There was a minute in between each trial, so the sessions lasted about 30 minutes. We measured sign tracking by a number of licks to the ethanol sipper, which was recorded by a lickometer. Goal tracking behavior was measured by the number of head pokes into the sugar pellet delivery site, which was detected by a photobeam sensor. While we were interested in the group differences in acquisition of sign tracking and goal tracking behavior, we were also interested in measuring how preference for ethanol changed with more sign tracking experience. And to test this, we conducted individual 20-minute 2-bottle choice tests for each rat in a separate testing room once a week. So finally getting to the results here. There's a nice graph showing the differences we found between housing conditions for sign tracking acquisition. We did see a significant main effective housing as well as a significant 2-day blocks by housing interaction. So what that tells us, and what we found in the post hoc test, is that the enriched group demonstrated greater lick rates than the standard group. We also see that the enriched group acquired sign tracking behavior at a faster rate than the standard group. So moving down to goal tracking acquisition, we found a significant three-way interaction between 2-day blocks, period, meaning pre-CS presentation and during CS presentation, and housing. We can see from the graphs that, interestingly, both groups demonstrated goal tracking behavior, since we're seeing head poking activity during the CS period. However, we see for the standard group that this consistent increase in CS head pokes and decrease in pre-CS head pokes, which shows greater condition goal tracking than we see for the enriched group. So as I mentioned before, we were also interested in seeing each group's preference for ethanol and whether that changed with more sign tracking experience. So in the first chart there on the left, you can see the numbers for those 20-minute 2-bottle choice tests conducted before sign tracking sessions began. On the y-axis is the preference for ethanol, and this was calculated by taking the amount of ethanol consumed and dividing it by the total number of both ethanol and water consumed, then of course multiplying by 100 to get the percentage. Prior to the sign tracking phase of the experiment, we had a pre-exposure period in which we started to allow the animals access to the ethanol solution, and we gradually increased the concentration from 3% up to 5%. So, and the numbers on the top of the bars in parentheses represent the average absolute volume of ethanol solution consumed for each group. So what we found here was that the enriched animals consumed significantly more ethanol at the 4% and 5% concentration, but the differences in preference between the two groups was not statistically significant. So looking at the chart for preference tests during the sign tracking part of the experiment, that's on the right, we see similar results. So while the enriched group did drink significantly more ethanol solution than the standard group, we didn't see any significant differences in ethanol preference. So in an attempt to get the rats to consume more liquid overall, we did introduce a postprandial method of feeding in which rats were food deprived overnight and then fed just prior to the preference testing to kind of induce thirst. We found that this method actually decreased ethanol preference when compared to ad lib feeding, and furthermore, we did not really find evidence of preference changing over time, since the differences in preference early versus late in sign tracking were non-significant. Okay, so what are the takeaway points here? First of all, we found that sign tracking does in fact still emerge when animals are tested in a home environment rather than a separate testing chamber. We found that, contradictory to the Beckman and Bardo study I mentioned in the introduction, environmental enrichment actually increased sign tracking behavior. And one of the reasons we may have seen this different result is that we extended the training period to 18 sessions, and the greatest differences didn't even emerge until block 4 or 5. So it seems as though with extended training, enriched rats do ultimately display greater incentive salience attribution to a bottle CS than standard house rats do. And maybe perhaps this is because this novel CS facilitates the object exploration behavior that may be more natural to the enriched rats. These are ideas that could possibly be explored maybe more by future research. And finally, although we did see some increased ethanol consumption during the 2-bottle tests, followed by the acquisition of sign tracking, we really did not see any increase in preference for the solution, suggesting that the liking of ethanol was not enhanced by sign tracking. This just further reinforces the idea that wanting and liking are two separate pathways that may happen to co-occur, but not in every situation. And that's kind of the overview of this experiment and what we found. If you do have any questions, you can drop them in the live chat or feel free to email me. Hopefully my email address should be available to you. So thanks again for visiting our booth, and I hope you enjoy the conference.